Welcome back to our last session dedicated to gene organization and chromosomes. In the last video, we have learned that DNA can be quite compacted and that chromosomes are most highly condensed during the M phase of the cell cycle. This is actually quite important because this allows us to have a closer look at the chromosomal configuration of a cell. And this is also useful in detection of chromosomal disorders. We will have a dedicated lecture on mutations later on, but for now it is just important for you to remember that actually those M phase chromosomes are a very useful tool because we can look at the arrangement of the full chromosomes, and the full arrangement of the chromosomes in a cell is called karyotype. In karyotypes, chromosomes are isolated, and this is what you can see on the left-hand side, and they are then digitally, using a PC and dedicated software, they are arranged so that they are matched with the homolog or partner chromosome, right? And those chromosomes can be then arranged in the karyogram, which can be analyzed. Homolog chromosomes are of the same size, they have the same shape, and carry the same genes. And each one is inherited from each parent. How this work, how this actually works in detail is something we are going to be having a closer look in our lecture dealing with meiosis. But for now, please remember that each corresponding chromosome is inherited from either the mother or the father. And we number those chromosomes in the human cell according to the size. So how does actually isolating of the chromosomes and basically arranging them for a karyogram actually work in a lab? So what we are doing here, we are isolating patient's cell and expanding them in a dish, in a lab. We can also add certain chemicals and certain signals to stimulate mitosis, to stimulate cell division. So those cells are now proliferating quite rapidly. We can incubate those samples for up to 3 days, and we can add another chemical to stop the mitosis specifically in the metaphase. And M phase is actually the phase we want the chromosomes to be in order to be able to analyze them using a microscope. We can then transfer the cells to a tube and centrifuge to concentrate them in layer. We can then add a fixative, isolate the chromosomes and put them onto a microscope slide. We can then identify and photograph the chromosomes and cut out chromosome pictures and arrange them into karyotype using modern image analysis software. In order to make them visible, we can also perform certain stainings specific for DNA. And one very classic example of such DNA-specific staining is the so-called Giemsa stain. If you stain chromosomes with the Giemsa dye, you will end up with very, very defined pattern on each chromosomes, because heterochromatic regions with DNA that is highly condensed stain more darkly in the G bending. And each chromosome, as I mentioned before, has a very specific bending pattern, and this makes identification of those chromosomes that are sometimes quite similar in size much easier. So we have those darker regions in the same places on the same chromosomes, whereas the lighter regions correspond as well. So this makes identification of chromosomes way easier. There is actually another more modern way of arranging chromosomes into karyograms, and this method is called FISH, or fluorescence in situ hybridization. This is a quite modern laboratory technique used to visualize where a particular gene or DNA sequence is located within a person's genome. And the FISH uses specific probes for defined DNA sequences. We can then use so-called fluorochromes, fluorescent molecules, and bind them to those probes, thereby allowing identification of certain DNA fragments. Because we nowadays know what genes are coded on which chromosome, we can then identify chromosomes that correspond in the probe using a technique called hybridization. So how does the typical karyogram in a human look like? The first obvious thing is basically that we have always 22 different pairs of autosomes and two sex chromosomes. For male patients, this is X and Y. X is larger than Y, and in females, we would have two X chromosomes, right? So in this karyogram, it's quite evident that we have 22 exact pairs, right? We have our 22 autosomes, and they do resemble each other fairly well. And we have also two sex chromosomes, namely X and Y. And this means that this person is male. So what are the chromosomal features we can identify on the karyogram? The first obvious thing is actually that we have two identical sister chromatids. And each pair of these chromatids is attached at an area called centromere, right? This is the first important chromosomal feature that you need to remember. Also, most chromosomes have one short arm and one long arm, and we are referring to them as a P arm for the short and Q arm the Q arm to the long one. And this is actually quite important for our future lecture dealing with mutations, so please keep this in mind. Those centromeres that actually divide those arms are primary constriction regions on the chromosomes, and this is basically based on the microscopic appearance. Each DNA molecule that forms a chromosome must contain a centromere. We must also have two telomeres and a replication origins. The replication origins are nucleotide sequences at which replication starts. Telomeres, in contrast, are caps at the ends of each chromosome, and they contain a very unique repetitive sequence called telomere tandem repeat, and we have quite a lot of them. Importantly, those caps shorten with each cell division, so our telomeric repeats are becoming shorter and shorter, and this is actually due to the replication problem we have discussed in one of our previous lectures. DNA polymerase can only read DNA in one certain direction, and this actually results in an overhang at the DNA. Most of our cells actually are affected by telomere shortening, and this is also one of the reasons why we age. Once the telomeres become critically short, essential information on the DNA or on the chromosomes becomes affected by these shortenings, and the cell undergo programmed cell death. However, few cell types are not affected. Those are stem cells and unfortunately also cancer or tumor cells. They can deal with these problems, and they are therefore potentially immortal. And the reason is because both of those cell types use an enzyme referred to as telomerase. In this short video, I would like to show you how telomerase works and what makes telomerase unique.
The ends of chromosomes contain regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of the telomeric repeat can vary from species to species. The repeat sequence shown here is found in the telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for a DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang on the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from tetrahymena, which is a model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes the DNA strand complementary to the RNA found in telomerase, and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end, and DNA polymerase fills in the vacant region. A short region at the 3' end will remain single-stranded. The end result is that telomerase will have added many repeat sequences, from a few dozen to several hundred. This prevents chromosome shortening.
This is actually quite important for few reasons, and the first reason is that this mechanism actually ensures that cancer and tumor cells are not affected by aging. They are potentially immortal, and this generates a huge problem in cancer treatment and therapy. The second reason is that this process technically violates the central dogma of molecular biology, which claims that DNA generates mRNA, but not vice versa. And this is actually what telomerase is doing. It brings an RNA primer or RNA templates, and based on this sequence generates telomeric repeats, so it generates DNA. The third reason is that telomeres are highly attractive candidates for dealing with cellular aging. And indeed, this has been discussed not just in Scientific American, but it's also targeted by many different companies at the moment. So, after we have learned how normal chromosomes look like, I would like to highlight very few chromosomal abnormalities we are going to be looking at in detail in one of our future lectures. So we can have numerical aberrations that are usually caused by the failure of chromosome division, and this results in cells with an extra chromosome or a deficiency in chromosomes. We can have structural aberrations, where loss of genetic material occurs. We can have rearrangement of chromosomal material, and we can have deletions, duplications, inversions, ring formations, and translocations. Some of those chromosomal abnormalities can already be detected in the karyogram. What you can see here is a karyogram of a patient suffering from trisomy 21, also referred to as the Down syndrome. And actually, the Down syndrome is quite common because around 775 babies are born with this condition each year in England and Wales alone. As symptoms, the patients have reduced muscle tone, leading to hypotonia. They have eyes that are slant upwards and outwards, small mouth with protruding tongue, and flat back of the head. So people with Down syndrome also vary in personality and ability. Everyone born with Down syndrome will have a degree of learning disability, but the level of disability will be different for each individual. Interestingly, the chances of Down syndrome increase significantly with the age of mother. If the mother's age is 20, the likelihood is pretty low, with 1 in nearly 2,000. However, if the age of the mother is 45, the likelihood or the risk increases to 1 in 32. So there is high correlation between mother's age and incidence of trisomy 21. Another example of chromosomal abnormality is trisomy, which affects the sex chromosome, and in this case Klinefelter syndromes. This occurs in males with 2 or more X chromosomes, typically XXY. And those patients have more than 2 X chromosomes, increased and they have several symptoms. They have learning disability, especially speech problems, and they are not progressing through puberty and have fertility issues. Turner syndrome is another example of chromosomal abnormality. In this case, one chromosome is missing. This results in shorter than average size of the person, underdeveloped ovaries, and lack of monthly periods and infertility. So as you can see here, chromosomes are really important and they play a very, very crucial role in life and death of all cells. With this, I would like to end the session and thank you for your attention. See you in the next session. Goodbye.